Welcome back, BioMonsters. Today we're going to be reviewing the important process of protein synthesis. To help us out, I wanted to introduce you to a brand new BioMonster, Mrs. Davidson. Say hi.
Hello.
All right, so we're going to be talking about the major steps of protein synthesis, and as we fill them in, you're going to do the same thing on your paper. All right, so let's go ahead and get started. The first thing that we're going to talk about is what is the first step of making a protein? So what's the very first step that everybody should know?
Transcription.
All right, so I'm going to go ahead and fill in transcription. You should do the same thing on your paper. So transcription is the very first step, and how do we start this process?
You take a segment of DNA and unwind it, and an mRNA copy is made.
All right, so I'm going to go ahead and add my mRNA to my second blank. Now this is all review from what we talked about in our last class. So using that information, you guys should be able to go ahead and fill in your stop and jot. Once you think you have the right answer, pause the video and call your teacher over to check it for correctness. So now that we've talked about an overview of transcription, and we know that for transcription we're going to use a DNA to make a messenger RNA, we need to actually figure out how do we do that. So if I'm given 1/2 of DNA, like I am here in this particular example, you guys know from our previous lessons that if you have one side of DNA, you can always figure out the other side, right, Mrs. Davidson?
Right.
And what rule can we use to help us figure that out?
The base pairing rule.
Exactly. So if I have an A on one side, what would be on the other side?
A T.
And if I had a G?
A C.
A C. So we're going to use that same principle, but we have one minor change. And in messenger RNA, what are we never going to see?
A T.
Okay, so we're going to make a little note here. So we're going to never have T's in messenger RNA. So anywhere we would feel compelled to use a T, what are we going to use instead?
A U.
We're going to use a U. So let's go ahead and see if we can do this using our base pairing rule. So if I have a T on this side, what's going to go on the other side?
A.
An A. And if I have an A here, what's going to be now on my messenger RNA?
A U.
A U, because normally if this were the other side of DNA, it would be a T. And anywhere I would want to use a T, I'm going to swap it out and use a U. So let's keep on going. We have a T, so it's going to pair to.
An A.
And then A pairs to.
U.
T to.
A.
G to.
C.
C to.
G.
All right, and then G.
C.
A.
U.
T.
A.
And then finally C.
G.
Excellent. So now that we've done one side, we want you to take this next DNA molecule and using our principles of transcription, figure out what the other side is going to look like. And when you think you have the right answer, call one of us over to check it for correctness. For our next big question, we're trying to figure out what does transcription actually look like. And so we're going to need to talk about one brand new term. And what's our first brand new term that we're going to discuss?
A codon.
A codon. So a codon is every
3.
Okay, so we're going to have every 3 bases, and a base is just one of the letters that we already talked about, on a messenger RNA, which codes for 1 amino acid. So it takes 3 letters to make 1 amino acid. So in order to start this process, we're going to look at our original messenger RNA, and here it is right here. And all we're going to do is we're going to create these chunks, or codons. So where am I going to put in my lines, Mrs. Davidson?
Every 3 letters.
Every 3 letters. So
AUC, draw a line. GCC, draw a line. AAU, draw a line. And then UTG would be your last codon.
okay, so looking at this strand, how many codons do I actually have?
4.
All right, so now that we have our codons marked out, let's go ahead and label them. So we have 1, 2, 3, 4. And we already know that every 3 letters or bases equates to how many amino acids?
1.
Okay, so how many amino acids do we have here, Mrs. Davidson?
4.
All right, so now we have to figure out how do we go from this to actually making our amino acids. All right, so now we're going to talk about the official second step. And what's the second step of protein synthesis?
Translation.
All right, so I'm going to go ahead and write translation in my first blank line. And this step takes place where?
At the ribosomes.
And that should make sense to everyone because the process of translation is the process of making proteins. And we know that proteins are only made at the
ribosomes.
All right, so in this step, the messenger RNA is going to be translated, but we need a helper in order to do that. And who's our helper?
tRNA.
Okay, and the T in tRNA stands for what?
Transfer RNA.
It stands for transfer. And what exactly is being transferred?
The amino acids are being transferred to the ribosome.
Okay, because we actually need those amino acids to grow this bigger thing that we need to make. And what is it that we need to make?
Proteins.
Excellent. So now, tRNA, the easiest way to recognize it is just think about the first letter. tRNA is actually shaped like a
T.
Actually shaped like the letter T. All right, so one side of my tRNA has something called an anticodon. And this term anti means the opposite. So it's the opposite of what we just talked about prior to on our messenger RNA. So the anticodon is going to be just the complement or the base pair of what we see on our original messenger RNA strand. So it's going to actually line up with the codon on our mRNA that we've already talked about on the previous section of our notes, okay? The other side of our tRNA now is going to carry the thing that it's transferring that we talked about before, and what is that?
The amino acid.
Okay. And if you wanted to take a look, here's an actual picture of our tRNA. So here's my amino acid at the top. The structure is shaped like a T, like we said, our tRNA. And then on the opposite side, we have our anticodon, which is just going to complement or base pair with our 3 letters that we see on our messenger RNA. Now that we've talked about that, let's look at our next question. Our next question says, what does transcription look like? So in the picture below, we want to label our messenger RNA, our tRNA, as well as our amino acids. And some of this stuff should be fairly simple because it looks like some of the things are already filled in. So let's start with our messenger RNA. Where do we see that?
At the top, but it shows you the 3 to 5 and has all the letters in a string.
Okay, and how do we know that this is messenger RNA and not DNA?
Because it's only single-stranded.
Okay, and what else do we see?
We see U's.
Okay, and that's an easy way. So we know it's single-stranded and we only see U's instead of T's, so that we know that right away this is our messenger RNA. The next thing that I'm looking for is my tRNA, and what shape is tRNA always going to look like?
Looks like T's.
Okay, so I have 2 here, so I'm going to go ahead and label this as my tRNA. And then the last thing that I'm looking to identify are my amino acids, and I have a bunch of them here, and I'm just going to label one. So here is my amino acid. All right. Now that we've done that, we're going to move on to our last thing, which can be the trickiest. We're going to have to figure out how to break the code, how to turn those 3 letters that are a part of a codon into an actual amino acid to grow our protein. So there are 2 types of codes that we're going, or 2 types of code breakers or charts that we're going to use to help us figure this out. So it says scientists have figured out which mRNA,
Codons.
okay, codons, code for which
Amino acids.
amino acids. So once we figure out how to use these 2 charts, there's never going to be a problem with figuring out what stretch of mRNA, what that stretch actually codes for. So let's see if we can go ahead and figure out how to do it. So we're going to start out with step number 1. So if we look down here, it says transcribe the DNA into messenger RNA. All that means is we're going to use our base pairing rule, but what are we never going to have in messenger RNA?
T.
Okay, so here's our first example. Our DNA is TAC, GGG, CT, CACT. So now all we're going to do is turn it or transcribe it into our messenger RNA. So using our base pairing rule, Miss Davidson's going to shout out what I'm going to write, and you need to write the same thing down on your paper. All right, let's go.
A, U, G, C, 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 G, A, G, U, G, A.
All right, so now that we've just done transcription, the next thing that we need to do is we actually have to break this up into groups of what?
3.
Okay, so I'm going to go ahead and I'm going to put in my line just like we did prior to in our former set of this set of notes, right? So how many codons do I actually have?
You have 4.
I have 4, so how many amino acids am I looking to make?
You're looking for 4.
Okay, so now what I need to do is I need to take this first group, so AUG, and I'm going to take it up into the chart. We're going to start with the circle chart first because it's a little easier to use. For the circle chart, we're always going to start in the middle. So our first letter, we're going to try to find it in the center of the circle. And what's our first letter?
A.
So A is right here. So here's my A. What's our second letter?
U.
So now I know because this is my first letter, my answer is going to be somewhere in this quadrant of my circle. So I'm looking for U in the next set of letters. So here's my U. And now I know my answer is going to be somewhere in here. And then what's my last letter?
G.
So here's G. So what is going to be my first one? AUG. What is it going to be?
Methionine.
All right. So I'm going to go ahead and use the abbreviation and write MET for my first amino acid in my protein. Now I need to find my second codon, which is CCC. And to find that, I'm going to go ahead and use my chart that looks more like a bingo card. And the way that this is going to work is I'm going to take my 3 letters and I'm going to label them 1, 2, and 3. I'm going to find my first letter, which is a C, along the left-hand side. So if I'm looking along the left, here's my C. So I know my answer is somewhere in this row. But that still leaves me a lot of possibilities, so I need to narrow it down. So I'm going to look at my second letter. My second letter is also a
C.
and I'm going to find that across the top. So looking across the top, here's my C, so I know my answer, I'm going to come down, is going to be somewhere in this box. Okay, so next thing that I'm going to do is I'm going to find my last letter, which is A.
C.
I'm going to find it across the side here on the right hand side, and here's my C. I'm going to go over, so here's where my answer is. It's CCC, and what does that code for?
Proline.
Proline. So I'm going to write the abbreviation right down here. So PRO. That's my second amino acid. Now I need to do this 2 more times. So now I need to find GAG. So I'm going to go back up. I'm going to use my bingo card version again. So I'm going to find G on the left hand side. Here's G. My answer is somewhere in this column. And then my second letter is an A. I'm going to find that up at the top. Here's A. That means my answer is somewhere in this section right here. And then my last letter is going to be a G. So I'm finding G along the side. Here it is, GAG. So my answer should be?
Glutamine.
There we go. So I'm going to write that. And then I have one more. And for our last one, let's go ahead and use the circle chart. So we have UGA. So looking for UGA, I'm going to find U in the middle. Here it is. So my answer is going to be somewhere in this section. Then I'm looking for what letter next?
G.
All right, so here's G. So my answer is going to be somewhere in here. And then my last letter is going to be?
A.
A. And then if I find that, notice what it says.
Stop.
So that's actually the end of my protein. So it's going to be methionine, proline, and then we also have glutamate. So now I need to take that and look at my chart for the trait. So I'm looking for MET-PRO-GLU. And when I look down here, MET-PRO-GLU, my trait is that it's going to have free earlobes. All right, so now that we've gone from DNA to messenger RNA, which is our transcription, and then messenger RNA to a protein, translation, and then we figured out our trait, which is our phenotype, we've done everything that we need to do, and we've utilized all the skill sets that are going to be necessary for the test. So we've done example 1 for you. Your job for your Stop and Jots is to do activity or example 2 and example 3. And when you think you have the right answer, call one of us over to check your answers for correctness. Thanks, guys.